Hi there, this is Dr. Dan Guerra, and this is Authentic Biochemistry. Yes, right, I'm going to be doing a video lecture today. It's been quite a while, and I've been feeling remiss in not doing it. And any rumors that have suggested that I've disappeared into the high country of the Idaho mountains are almost largely unfounded. So I'm here to show you that. Right? And what we're going to do today is have a lecture on membrane lipid components associated with epitranscriptomic modification that is linked to a certain form of prostate cancer. And as soon as I get this going, we will get it started. All right, now there's one modification I want to make here. And it's up here. Sorry, I'm trying something new here, and it's a little bit more technical than what I normally do, but it's fun, right? It's fun. So I'm going to do this. You reach right over here and do that. All right. Now, I'm calling this Caveolae membranes and extratranscriptomic cargoes in cancer. All right. And we're going to leave it at that particular name. And we're going to start talking directly about a paper that was published just late last year in Clinical Translational Medicine. And this paper is going to be talking about those membrane structures that I spent some time on over the last year when we were doing aging and then when we did diabetes. So these are membrane structural components, which are a structure that is associated with membrane lipid rafts, but which becomes a permanent structure in the plasma membrane once it's fully assembled. Now, by permanent, I mean that it becomes a fixture of that plasma membrane. But at the same time, because it's a raft, it can mobilize. And because it can mobilize, it can sometimes generate vesicles. And these vesicles can transport extracytoplasmically and indeed extracellularly components of the cell. And some of those components are microRNAs, which are going to then, of course, change gene expression to whatever the specific target messenger RNA is once that extracytoplasmic vesicle enters a new cell, where that microRNA can then be introduced, and then because of homologous pairing, that is complementarity, that microRNA will bind to its complementary messenger RNA and then change its expression, typically stop its expression at the translational level, right? So I remind you that that's an epigenetic phenomenon. Right? Okay. So as we say here, caveolae are flash-shaped subtype of a membrane raft. So again, there's multiple subtypes when you look at them structurally. And it's formed by the interaction of a structural membrane protein called caveolin-1, CAV1, and another protein, which sometimes people get confused with. It's a cytoplasmic protein, cavin-1, and that's called cavin-1. So you have CAV1, cavin-1. One's typically in the membrane, that's CAV1, and the other one is in the cytoplasm, but still interacts, okay? So I'm sorry about the way the nomenclature can be a little bit tricky at times, but that's how it is. Now those raft structures, as you know, are enriched in cholesterol and in ceramide, right? That's how those rafts form, and the endocytic compartment, that's how they transfer from the endoplasmic reticulum to the Golgi apparatus to the plasma membrane, and then back into the cell. Right? So now, for this particular paper, we're told that CAV1 and cavin-1 are translated, of course, and trafficked to the plasma membrane. in healthy human cells. Right? There's nothing unusual about that. Those are, in fact, components of how caveolae are generated, and they just happen to be cargo, or that is, they are parts of the structure of the membrane lipid wrap at the same time. Because they're polypeptides, they're also cargo. Now, it turns out advanced prostate cancer cells that have been isolated from human disease patients express CAV1, okay, without cavin-1. All right? So the two-component protein system, protein-protein interaction, is lost. Now that apparently leads to dysfunctional pro-metastatic non-caveolar CAV1 membrane raft domains. Okay? So that's what happens because cavin's gone. Right? Now, that could allow you to generate a null hypothesis for experiments, which is what these people did in this paper. Whereas CAV1 and cavin-1 normally provide cargo transport function for extracellular vesicular trafficking, the loss, we call that EV, the loss of that protein-protein interaction within that membranous system may be involved in prostate cancer. The reason we could make the hypothesis is because of what the observation was, right? So there's a genotype then, which is CAV1 plus, cavin-1 minus. That's the genotype, right? Cavin-1 is no longer being expressed. And that's observed in non-caveolar PC3. It's just a reiteration of what I just said. PC3 is aggressive prostate cancer. It's a particular cell line. It's kept in vitro and used for these kinds of studies. So when you do an ectopic expression of cavin-1, you introduce cavin-1 back to these PC3 cells. So now you've got the genotype CAV1 positive, cavin-1 positive, right? And they're going to be called, and throughout this paper, PC3 cavin-1, because you added that gene back. Not only do you get a rescue of the caveolae formation, that occurs, but it attenuates multiple oncogenic properties, both in vitro and in vivo. So that's really interesting. So that means, you can write, basically from the hypothesis, that cavin-1 seems to be normal physiology, and the loss of cavin-1, however that occurs, leads to at least some phenotype linked to caveolae-associated prostate cancer within the cell lineage worth being studied. Now, besides suppressed growth and migration, the PC3 cavin-1 cells display an altered tumor, such as reduced stromal myofibroblasts and an attenuated metastasis that goes to lung and bone. These are obviously animal studies. So the overall phenomenon then seems to be linked to an altered EV, right? Those are the extravesicular vesicle system content, which correlated with changes in the osteoblast proliferation, which is a component of the cancer, and then differentiation, ultimately leading to a tumor. Now recall that these vesicles, these EVs, are surrounded by their own membrane, and they're released through the caveolae from a cell. So that means we have to talk about their transport mechanism and their biogenesis, right? So EVs transport messenger RNAs. This has been known for a long time. It's interesting when you think about it. A cell goes through transcription and then processing of that transcript to the cytosol or maybe to the endoplasmic reticulum. And then that messenger RNA, rather than being translated in that cell, is mobilized into an exosome and transported out of the cell, which means that whatever is expressing there is at an RNA level. I think you can consider the implications of this, right? So you get the messenger RNA in these vesicles, but you also get microRNA. And remember, those are short RNAs, which are complementary to messenger RNAs, sequence level. And they have a certain motif, which I'm going to mention in a minute. And with that motif and that sequence complementarity, that microRNA will find a messenger RNA and will prevent its translation. And that is an epitranscriptomic phenomenon. And therefore it falls into the epigenetics lecture. This one being number 20. So the two major routes of these EVs being defined based on the route of release. Exosomes are generated in the endocytic system in multivesicular bodies called MVBs, and also they're released via fusion, and to continue, ultimately they're released via fusion of the delimiting membranes of that MVB with the plasma membrane. Okay, so you have these exosomes that are moving through the endocytic compartment that bind, that fuse to the plasma membrane as an MVB, a multivesicular body. And when they do that, they're found in the plasma membrane and then they can leave. But there's another route. Larger microvesicles can just extravasate. So that means these vesicles are formed within the plasma membrane because of the lipid component that allow them then to mobilize. What's that lipid component? Cholesterol and ceramide have specific molar concentrations, ratios. And those molar concentration ratios also are dictated by the type of ceramide. I remember there are multiple forms of ceramide because you have 6 different isoforms of ceramide synthase, and each one of them have different roles adding fatty acids to that amide linkage. So there's a great deal of heterogeneity in the membrane lipid raft. And that is related to the cargo and what are the two mechanisms they go by where they become extracellular. Okay. So the term exosome is used when the mode of biogenesis has been established, whereas you call them EVs, and that just denotes a mixture of vesicles, and we don't really know the mode of the biogenesis. And in this study, they're calling them EVs because they didn't study the mode of biogenesis. Okay, that's what they're telling you. But there is a mode, and there is a specificity, and I just explained it to you, partially related to ceramide molecular species, which is then related to ceramide synthesis. And that means fatty acids, and it's going to come back later. Now, specific fatty acids, right? Linked to that amide, linked to that nitrogen atom in the sphingosine base. Now, both vesicle-based communication systems follow well-described transport processes in normal physiology, of course. But when they're corrupted, you find them in cancers and sometimes in other non-etiologically derived diseases. So where we don't really know how they form, but they're found in some diseases that don't seem to be related at all to this mechanism or to, say, cancer. But the diseases of being non-etiological, what I'm trying to say there, these are diseases such as cardiovascular disease, autoimmune disease, microenvironmental within the system, not related to an etiologic agent such as a bacterium, a fungus, a parasite, or a virus. That's a different mechanism, okay? Particularly with all of those potentially endocytic pathogens. And we've talked about that in the past, but it's been a while, but that's not what we're talking about here. This is all normal physiology, right? Packaging RNAs into these endosomal, exosomal trafficking systems and moving them out. But they can become pathophysiological and pathobiochemical when something's missing or something's corrupted in the system. And here this Caveolae is missing, remember. So ultimately, the pathological or physiological effects delivered by these vesicles are largely dependent on the cargo. Now that makes sense, because the cargo is going to give some kind of resplendent specificity to what's happening with that exocytoplasmic vesicular trafficking. Not just a lipid, but whatever's in it, usually protein of some very important significance. Now, Epitranscriptomic modify the expression of several genes. We know this. By directly binding to messenger RNA to suppress protein translation. So when you have epitranscriptomic microRNA, they'll modify the expression of genes depending on the sequence complementarity. In PC3, remember that's the prostate cancer cell line, EVs, microRNA modify, as we have been showing you because we told you the pathophenotype, an osteogenic activity in the prostate system. Osteogenic, we talk about the microRNA species, miR148A3P, that's a specific molecular signature, was reduced in Caveolae PC3 EVs. Remember the Caveolae PC3 is when we've added back Caveolae. So we are losing the tumor phenotype, right? We're losing the tumor phenotype because we added back the Caveolae. And they're telling you that they get a decrease in that microRNA when they make the cell line now less metastatic, which means less osteoblastic, right? So that's interesting. So Caveolae then, its expression reduces the microRNA that comes up into those vesicles. But it doesn't alter total cellular levels of Caveolae expression. It alters the PC membrane raft and the EV proteome instead. So rather than seeing a high elevation of Caveolae in the cytoplasm, you don't see that, even the ectopic expression of it. It all seems to be associated with this interaction, yet very interestingly, whatever mole percent of that Caveolae is necessary, it doesn't end up, right, in the EV, okay? Which is actually known because that would be also probably pathophysiological and therefore underpinning pathobiochemical. So Caveolae indirectly regulates extracytoplasmic vesicular microRNA content. That's what they're telling you. So the mechanism still needs to be totally worked out, but obviously it's going to be a lipid-protein interaction. Now, protein cargoes are loaded to these EVs, as we, classically, canonically, via endosomal sorting complexes required for transport complex and tetraspanin-based mechanisms. Now, this has to do with a whole description of proteins that we went through before. We're not going to get into it now, right? Because it took me about, I think, 2 lectures to go through that sorting mechanism, the SNAP proteins and all that. The synaptosomes, remember all that? The snaposomes and the accessory proteins, right, that are associated with this tetraspanin protein, which is like a cytoskeletal protein within the membrane raft. At any rate, we're not going to talk about that. I'm just telling you that's classically what happens. The EV membranes are enriched in cholesterol and sphingolipids. That sphingolipid is going to be ceramide primarily. And EV membranes enriched in those lipids are also how caveolae come out in terms of their lipid composition. And we talked about this. That's why caveolae are detergent-resistant membranes, because detergents don't break down those highly neutral, highly hydrophobic lipids. You have to have other kinds of reagents to corrupt those membranes. Those membranes are very sturdy. Remember, we were talking about caveolae being important in fatty acid transport and how they were involved in type 2 diabetes because they were associated with insulin resistance. That's right, go back and listen to those lectures. I talked about that in the aging arc and also in the type 2 diabetes arc. So see, this stuff comes back to us. Every once in a while, you do get reminded or refreshed of something else in a totally different pathophysiological situation. But the mechanisms are not different. But the players are, of course. So you can't make models of this. You have to study it empirically each time. And the more you study empirically, the deeper you must go. This is how research science is supposed to work. Now, rafts are often enhanced. Okay, so you get these detergent-resistant membranes. That's what caveolae are. And that's what membrane lipid rafts are because of their lipid composition. Okay. Rafts are often enhanced in properties like increased or enhanced bilayer thickness and an increase in viscosity. These two components are related directly to the high hydrophobicity of the lipids involved. You see. And also the unusual structure of cholesterol, a cyclopentanoenanthrene ring, makes very interesting deposits within a membrane bilayer. And we talked about that too when we did straightforward membrane biochemistry, biophysics. In fact, we're going to see that start of that today. Now, anyways, what that does, increased thickness, increased viscosity, it then generates essentially a cavity in the membrane, or a cave, caveolae, right? And that would support what? Functionally, a sequestration of what? Cargo. And that sequestration then ends up being a compartmentalization. So you have micro-compartmentalization within membranes because of the lipid structure. That's the point. Now, due to these biophysical properties, caveolae sort the membrane raft proteome. That's a very, very important statement. How do you get the proteome sorted? Right? It's the lipid transport mechanism, which is related to the lipid composition, which is ultimately related to the fluid dynamics of the lipid-lipid interactions, which makes it a biophysical property. Now, it could be argued that this Caveolae regulates microRNA loading to the EVs, and perhaps certain RNA binding proteins specify that extravesicular microRNA loading. Okay? So you've got Caveolae, which maybe is involved in RNA binding or loading, but we also know there are proteins that are specifically just RNA binding proteins. That's right, and here we go. Final component of this very short description of this paper. The heterogeneous nuclear ribonuclear protein K, HNRNPK, actually mediates the CAV1. Remember, that's the caveolin 1 protein that's found in the membrane naturally. It's not the cytoplasmic one, the cavin. HNRNPK mediates CAV1 dependent sequence specific microRNA sorting into the caveolae and also into these vesicles. Thus, exosomal HNRNPK and its direct interaction with the microRNA, because that's a binding protein, right, may be linked to prostate cancer by processing the microRNA via membrane remodeling, retailoring, driven by the non-caveolar, because remember that's not actually a caveolar protein, CAV1. So you need to keep all these proteins and these lipids in mind. You need to sort them out just like the cell does. And that's what I've done in this sentence. So now this comes from the paper. And let's just take a look at it. You've got non-caveolar CAV1 protein. There's a caveolae that stimulates membrane changes, including these multivesicular masses, okay? The bodies, they're called. So the multivesicular body then has CAV1 altered rafts. Now that raft now can pick up this specific RNPK that has this unusual nucleotide pattern. Okay? That's the ASUGNA motif containing microRNA. So it's going to pick up that microRNA because that's what the HNR NPK recognizes. So it's a protein binding to that motif, that nucleotide motif. You see? You see, it's got a uracil, right? And a guanine, and then any nucleotide, then an adenosine. Right? That's what we're talking about. So non-caveolar caveolin 1, that's CAV1, drives non-canonical localization of the HNRNPK, that polypeptide. It's coming from the nucleus. Not sure how it's shuttled, okay, but it's shuttled from the nucleus because normally it's in the nucleus because binding to RNA. Here it's out here binding this microRNA. Maybe it transports the microRNA. Okay? At any rate, takes it to these bodies, these multivesicular bodies, which are CAV1 altered because they don't have cavin interaction, right? Missing in this cancer cell line. So MVB localized the HNRNPK mediated EV loading of that ASUGNA motif containing microRNA, which is required for EV induced in vitro. This is what happens when you get it in vitro. You have that RNA, you get in vitro osteoclastogenesis. That is a component of the metastatic delivery of these exosomes because it's got that microRNA. It induces that. There is a link between non-caveolar CAV1 driven membrane remodeling, okay, that's what's going on here to make these MVBs, and the exosomal microRNA composition, specifically this microRNA, the one we just mentioned, right? That 148. And this link is a motif-guided microRNA recruitment via the protein that binds to it, the HNRNPK, because it gets redistributed along with the microRNA into the MVB. That's because of CAV1 and CAV1 alone altering the RAS because you don't have CAVin1, the cytoplasmic component. You get that? All right. I know you do. So there is a link between non-caveolar CAV1 and that whole redistribution. So the functional role of RN, P and K in the PC3 EV, now that's the disease, that's the prostate cancer cell line, PC3, EV-mediated osteoclastogenesis, and EVHN RNPK detection in metastatic prostate and even shown in colorectal cancers implicates the extracytoplasmic vesicular HNRNPK as a potential biomarker if you find it in circulation in metastatic cancer. This is 2021, late 2021 paper. So this is, again, a clinical biochemistry type of paper. So where do you think this work is going to go? It's going to go directly to the pharmaceutical researchers to figure out whether or not they can work something to alter the mobility of that polypeptide. Right? Now, RAF disruption by two different systems. I'll tell you what they are in a minute. M-beta CD, I'll explain what that is, or adding any omega-3 polyunsaturated fatty acid will also reduce the HNRNPK cytoplasmic localization, and that'll corrupt the entire metastatic phenomenon. Because if you don't get our HNRNPK into the cytosol, it won't be carrying with it the microRNA, and that microRNA is associated with the cancer, with osteoclastogenesis, which is a phenotype of the cancer. So RAF disruption by using this compound or any omega-3 fatty acid, which is polyunsaturated, that would mean something like eicosapentaenoic or docosahexaenoic, maybe docosapentaenoic of the omega-3 positional isomeric form, reduces RNPK, cytoplasmic suggesting pharmacotherapeutic potential. Put a question mark there. Yeah. Now, I will tell you a little bit about these cyclodextrins. So that MCB that we were just talking about is a methyl beta cyclodextrin. It's a carbohydrate. And preferentially what it does, the reason they used it, and the reason why it worked to get rid of that HNR and PK, is because it preferentially removes cholesterol from the liquid disordered phase in giant unicellular vesicles. Now, that was first published in this membrane biology paper of 2011. I dug this out for you so that you know how methyl beta cyclodextrins work. Again, remember, this is authentic biochemistry. So I don't mess around. You want to know what this does? I'm telling you what it does. There's going to be a little twist to this in the end. There always is. This is the original paper where they were looking at what this compound did, this carbohydrate. So it removes cholesterol from liquid disordered phase in giant vesicles. This is all an in vitro study, obviously. So giant unilamellar vesicles, or GUVs, made of 1,2-dioleophosphatidycholine, 1,2-dipalmitoylphosphatidylcholine, and free cholesterol in a ratio of 1 to 1 to 1. These are just glycerol lipids necessary to make the vesicle. And, they're pretty common, dioleo PC and dipalmitoyl PC, okay? But this is how they made the GUVs, and it had to be a three-way split to make them turn into unilamellar vesicles, because they also added cholesterol at a 1 to 1 to 1 molar ratio. And they did it at 27 degrees, and it presents with cholesterol distributed into the LO and LD. That's the ordered liquid phase of the membrane, and the disordered liquid crystalline phase of the membrane structure. These are good lipid people, membrane biology paper. The two phases of that membrane lipid component, biophysical phases, can be distinguished and visualized using Lordan generalized polarization. This is an optic sort of method associated with a 2-photon excitation fluorescence microscopy. This is just naming you one of the techniques used in membrane biophysics and biochemistry. Thus, M-beta-CD removes cholesterol preferentially from the more disordered phase. Now, why is that interesting? Because a disordered phase would be something like a membrane lipid. Disordered enough to become mobile, you see? Now, they didn't know this, 11 years ago or 10 years ago when that last paper we're talking about was published. They didn't know it was going to happen 10 years later, but we do. That's the beauty of reading the scientific literature. It's all there, but you have to read it, and you have to kinetically examine it, then dynamically organize it to synthesize the information. First an analysis, then the synthesis. Platonic metaphysics. Now, and also epistemology, of course. Now, the process of selective cholesterol removal is dependent on the concentration of the M-beta-CD. Okay, good. So that means it's titratable. Now, here's something interesting. At high concentrations, the M-beta-CD also removes those phospholipids. Now, if you carefully look at that paper published in 2021, I didn't see in their materials and methods where they were doing multiple concentration mediated alterations of those vesicles with their M-beta-CD. So I don't know if they were using really high levels. If they're using really high levels, they would have altered that membrane beyond just the cholesterol content. But if they were titrating for cholesterol removal, and I think indeed they were in that 2021 paper, then they probably avoided this potential confounder. It's a potential confounder. Because then you have to explain more what's going on with the phospholipids. Okay, so that paper we didn't quite complete. I gave you enough of it for you to understand why I wanted you to hear about it, because we basically were talking about cytoepigenetics. And this is lecture 20 done in the video, right? So I didn't want to end the lecture with what I did in number 19, which of course was, I think, pretty dynamic and pretty interesting on its own right. I wanted to cap it all with a video lecture. So I wanted to make sure that we're still epigenetic phenomenon, and that paper described that, even with a pathophysiology, a specific cancer, very lethal cancer in males, a prostate cancer. So it's relevant clinically. So therefore, relative to biomedicine, which I like to do in my lectures. But also it allows me now to move on. So this is one of those lectures where we started off talking about cytoepigenetics. Now I'm going to discuss a general lecture in membrane biochemistry. And then from there, we'll launch into a new topic. So I'm being very crafty here in making this lecture, and specifically did it as a video to do exactly that. So I could use the video as a drama sequence to be able to move from one topic to the other. So let's talk now about membrane function. This is more just straight membrane physiology, membrane biochemistry. So we're pulling out of the cytoepigenetics now. Okay. Membrane function, they serve as semi-permeable isolating barriers to separate contents of cells from external environment, or pathway constituents within organelles to coordinate homeostasis. And we talk a lot about this. We just did the last 20 lectures, right? Now, proteins in cell membranes, of course, have multiple functions. And typically, when people talk about membrane biochemistry, they talk about the proteins. Because I'm a lipid biochemist, I talk about the proteins because that's necessary. It's a little bit about, slumming it a little bit, I suppose. But I really want to get to talk about lipids. And so I bring in the proteins so that I capture people's interest. But then, of course, we get into lipid biochemistry. Because it's the more significant and far more interesting. Because it's more complex. And therefore more challenging. And what's more interesting than that which is challenging, right? So, okay. Proteins in the cell membrane can be transporters of substances across the membrane. Hello, that caveolin discussion we just had. And that binding protein we just talked about. You can get enzymes that catalyze biochemical reactions, like what? Sphingomyelinase, GTPase, ATPases, receptors on exterior surface that bind external ligands. Here you're talking about classical hormones, growth factors, things like that. Insulin receptor, glucagon receptor. And then you have mediators that aid ligand receptor complexes in triggering a sequence of events. This would include things like adenylate cyclase, guanylate cyclase, right? G protein coupled receptors to the production of secondary messengers, to I just described to you. Inositol pyrophosphate, and inositol phospholipids, and generating IP3 and then diacylglycerol, for example. That's another secondary messenger system. So anyways, all of that will alter metabolism produced inside the cell, but can also have effects outside the cell, depending. So plasma membrane has selective permeabilities. So you have channels and pumps for ions and substrates, right? Glucose uptake, sodium, potassium uptake, things like that. Calcium flux. Specific receptors for signals. Those would be things again like insulin or estrogen or testosterone. Right? Exchange materials with extracellular environment, and that we just went through. Exocytosis and endocytosis. We went through it as a very specific clinical discussion of a specific cancer and brought in the epitranscriptomic signature of that cancer via discussion of exocytosis and endocytosis. Now, when I was talking about aging, I pulled this slide from that aging literature. Skeletal muscle contraction in chronic myopathies is involved in aging, part of the sarcopenia, but also cardiac muscle contraction, acute injury, such as ischemia reperfusion injury, that could be associated after heart failure or an ischemic attack. Pore-mediated injuries, all these are associated with an increase in aging and corrupting membrane integrity. Right? All of this is pathophysiology. Pore-mediated injuries triggered by immune cells, by diapedesis, for example, of macrophages forming foam cells. Extracellular matrix proteases, we spent a lot of time on those. Autophagy, of things like peroxisomes and mitochondria. And of course, apoptosis and various forms of necrotosis, ferritosis. Right? Neuronal membrane injuries also occur during physiological aging and both acute and chronic illnesses. So some acute brain injuries, obviously, and then the neurodegenerative diseases, Alzheimer's, Parkinson's. Right? Migration-induced injuries in immune cells and fibroblasts under physiological conditions and in cancer cells during the whole invasion metastasis process. That's also going to corrupt membrane integrity. So right away, I told you a few basic things about membranes, right? Now I'm telling you what happens in the normal process of aging. Does that mean when you get really, really aged, this is the only time this happens? No. It means that whenever the cell ages, and cells start aging soon as they divide. So that means that the cells are always aging, and the whole tissue is aging, and the whole system is aging. Now, of course, you can get cellular replenishment via mitosis, right? And you can also get cellular differentiation, of course, from stem cells. But the point I'm making is that when you talk about membrane activity like we just did with the caveolae and the microRNA and the HNRNPK and the interaction between those proteins and how you get a microRNA extra cytoplasmic and therefore extra cellular, so that that microRNA can traffic to other cells and thus apparently carry with it that osteoblastic oncogenic event generating a metastatic cancer to the bone, for example, maybe to the brain or the heart. You can see how membrane corruption, which can occur all the time, has to be immediately associated with membrane repair, which means de novo fatty acid biosynthesis, which means fatty acid desaturation and elongation to the polyunsaturated omega-3, omega-6 fatty acids to maintain membrane integrity. And it also, of course, involves membrane turnover because of oxidative metabolism, such as the production of peroxy fatty acids and hydroperoxy fatty acids. And that's where the peroxisome comes into play as a very vital organelle. So I'm going to stop here because all I want to do is get to this point, and to give you that end of that cytoepigenetic phenomena that I thought was really important for us to discuss. So let me get out of here. Okay, we're going to have to move down here, I guess. I couldn't move that. Couldn't get my mouse to move, but I do it myself. Manually. All right, so let's get to where we can say our sign-off sequence here. All right. So this is Dr. Dan Guerra from Authentic Biochemistry Studios, a new studio. And hopefully you enjoyed this video lecture. Please comment on it when you see it on YouTube. I'll put it on YouTube. And let me know what you thought of it. And let me know if you want to see more video lectures or if you're satisfied with the audio sets that I've been doing in Authentic Biochemistry. I'll do more video because I'm set up to do it now, as you can see. But I'm not going to spend the effort to do that if you feel like the audio lectures give you just as much information and you don't have to see me delivering it. But you saw me, and now you know how I deliver the lectures when I'm doing them off camera. It's the same thing. All right, so let's do this. And I'm going to stop sharing this. I'm going to say my sign-off. Dr. Dan Guerra saying from beautiful inland Pacific Northwest of the USA. Bye for now.